So now we're going to talk a bit about the properties of membranes and specifically how they're able to maintain chemical gradients. So we saw in one of the previous videos that the membrane is fluid, so it's able to move. Now when we're talking about fluidity, the lipids and the proteins that compose the membrane are able to move relative to each other. So they can move like up and down, they can shift. As they're shifting, some molecules are going to be able to move between neighboring phospholipids to get to the cell. But which ones are able to do that is going to depend on their chemical properties and their size. Specifically, the fact that we have all these non-polar fatty acid tails, remember we have a double layer of those when we're talking about a membrane bilayer, that's going to prevent anything with a charge from getting through the membrane because it's going to be repelled by that really thick hydrophobic layer. So ions and large polar molecules are not going to be able to squeeze through the membrane because they're going to be repelled by that hydrophobic layer. So we can sort of rank which molecules are able to get across the membrane in order. So the cell membrane is permeable too. Permeable means that things can cross it. Gases and small uncharged molecules to a very high degree. So things like oxygen, nitrogen, and carbon dioxide gas can get across the membrane super easily on their own. They can fit between those phospholipids. They don't have a charge, so they're not repelled by that thick layer, that thick hydrophobic layer. Water is also quite able to get across the cell membrane, and there are some additional sort of ways the cell has of letting water pass through even more easily, which we're not going to get into in this course, but know they exist. Water is very able to get across cell membranes. But then we get to the things that aren't really able to get across. So remember that sugars have a lot of hydroxyl groups. They have a lot of oxygen. So they're both going to be slightly larger than these other molecules, and they're going to have those slight charges accumulating on them. So they're polar organic molecules, and they really aren't going to be able to cross the membrane without help. Same goes for ions. So ions, remember, are what happens when we have an ionic bond that basically dissolves in water. We have the two pieces come apart. And larger charged molecules and really big macromolecules have the hardest time getting across. So because of those strong charges when we're talking about ions, and really because of their size when we're talking about things like DNA. This is actually really convenient for us because the membrane can kind of select which molecules then of those larger ones are going to cross at which times. So we refer to the cell membrane as selectively permeable, and that means that some things can cross and others can't. This allows us to take in essential molecules, so things like oxygen have a really easy time crossing. So things that your cells need can get in. Things that are sort of intermediate parts of cell processes that are still going on can stay inside, but we can get rid of waste products. We're going to go through the different ways that we move things across the membrane in the next couple of videos. But for now, you should know that the word for this is transport. So transport is moving things across a cell membrane. And there are going to be two types: passive, which doesn't require energy and happens pretty naturally, and active transport, which is going to require energy in some form. However, in order to distinguish which we're looking at, passive or active transport, we first have to talk a little bit about gradients. So a really useful property of membranes is the fact that they can regulate what goes in and out and when, allows them to maintain what's called a gradient, which just means that we can have a lot of a molecule sort of concentrated up on one side of the membrane, and then a small amount of that molecule sort of loosely dispersed on the other side of a membrane. So usually this wouldn't happen. If there was no membrane, they'd all kind of spread out equally until they were evenly distributed, which we call reaching equilibrium. But because we have this barrier, and this barrier controls what goes in and out, we can have a difference on either side of the cell. So we call this difference in concentration, or later when you get to future courses, you'll see that we'll also think about differences in charge of the fluids on either side. But for now, we're thinking about concentrations. We call this a transmembrane gradient. So there's a transmembrane gradient if we have a higher concentration, so more of something, on one side of the membrane than on the other. When we talk about these gradients, it's helpful to name what type of gradient we have. So there are some words that just refer to where you have the concentration of the solute. So remember that the word solute means something that's dissolved in water. So that's our little particle dissolved in the water. In this case, it's our little red molecules here. Okay, so we call it the relative tonicity when we're talking about the concentration of something outside the cell relative to inside the cell. So here we have our cell membrane. Here's the inside of the cell. Here's the outside of the cell. Okay, so if we have the same amount of solute outside the cell as inside the cell, we call that an isotonic solution. That's because iso means same, so it's the same on the outside as on the inside. We can have differences though. So if we have more of the solute on the outside and less on the inside, we call this a hypertonic solution. Now, hyper means more, so like, you know, when you're feeling hyper, you're extra active. When we have a hypertonic solution, we have extra solute on the outside. And the opposite, a hypotonic. Hypo means low. So in a hypotonic solution, we have a low amount of solute on the outside and a greater amount on the inside. So I said before that all else being equal, things would just spread apart if they weren't blocked by the membrane. So if we have this example, and we can see that we have a concentrated collection of dots here, we'll think of this as our membrane. If it is able to get across the membrane, it's going to spread evenly across it. So these little blue dots will be able to travel through these pores or these areas in the membrane. So this line doesn't present a sufficient barrier to these molecules. So think of them as oxygen, for example, and this is your membrane. Oxygen can get across, these dots can get across. They just spread out evenly. We call that diffusion. And that's a natural process. It's like a law of the universe. Diffusion is going to be our first and easiest type of passive transport. So in diffusion, what you're going to look for to determine whether you're looking at a situation in which diffusion is happening, you're first going to identify where you have a gradient. So where you have a lot of something, where you have a high concentration, and where you have only a little of something. So where you have a low concentration. If you see something moving from that area of high concentration to the area of low concentration, so that then they're more evenly spread out afterwards, you're probably looking at diffusion, and you are definitely looking at passive transport. So this process is going to stop once we reach equilibrium because we no longer have a gradient. It's just the natural process of everything spreading out until it's equally distributed, but sometimes that means that a molecule is going to move from one side of the membrane where there's a high concentration to the other side of the membrane where there's a low concentration in that process of spreading out. So only small and nonpolar molecules are going to be able to directly diffuse across a membrane.